But I hope that today I can convey basically what you need to know as a non-expert. So specifically as a non-expert interested in robotic phenotyping, image analysis, and related PhenoRob activities. So I thought I would go over basically what is plant breeding to start with. So what do we define this? And then some of the things I think are probably the most important to understand as concepts. So what is a heritable trait? So what is heritable? What is a trait, etc. And then genetic and environmental effects, which I think is very important, especially for field experiments. And then I want to also talk a little bit about what plant breeding research involves. So the different fields that encompass plant breeding research. I'll have a little bit about what we're aiming to do with robotic phenotyping data, but I think this can maybe also be a major discussion point. So I'd be happy to answer questions about this afterwards. Okay, so what actually is plant breeding and what does it aim to do? Definition of plant breeding is actually just the genetic improvement of crop plants. So the key word here is actually genetic. So genetic means we are looking specifically at heritable traits, the improvements which can be passed from parents to offspring. We want to make changes or make selections that allow us to improve our crop plants permanently, basically. So the next generation should be better than the previous generation. How do we know what's a heritable trait? So the definition here of a trait is actually just any specific characteristic of an individual, or so something that we can observe. This could be anything from leaf size to plant height, etc. And heritable means it's passed from the parents to progeny, so it's not just a product of the environment. To give you an example of this, this is a group of random people. And we can see in this picture that there is variation for firstly clothing color, secondly hair length, thirdly hair color, and fourthly skin color. So clothing color we know is 100% environmentally determined, as is hair length. So you can effectively choose what color clothing to wear, and we know when we look at this picture, okay, clothing color is not a heritable trait. Hair length also, and I specifically looked this up, most people can potentially grow their hair as long as they like, if they can be bothered. So this is environmentally determined and not really a genetic factor. Hair color is hypothetically genetically determined, but we know some of the products' hair colors in this image are also environmentally influenced. And the same with skin color is largely genetic, but also depends on sun exposure. So this is relatively easy for us if we have an understanding of humans in general to work out which traits are heritable in this image that we're looking at. So which of the individual characteristics that we're looking at are ones where we would expect it to be inherited from the parents to the progeny and which ones not. So with humans, it's pretty easy. So we know a lot about ourselves. But when we're looking at the inheritance of traits in crops, this can be a little bit more complicated. It's not necessarily the case that we can simply look at variation and know which traits are heritable and which are determined by the environment. So these are some promising bioenergy or multi-purpose crops. And you can see there's quite a range of different phenotypes, so different the plants look very different in terms of their traits. And in each case, we need to work out which parts of the what traits are environmentally or genetically determined, or what the percentage contribution is. So can we simply look at variation and know which traits are heritable? No, we have to experimentally validate whether what we see is a product of genotype or the environment. And this brings us to a very classic and very simple equation, which is that the phenotype, which is what we see, so the characteristics of an individual, is a product always of the genotype, so the underlying genetic factors, which are heritable, passed from parents to progeny, and the environment, so the influences of the surrounding conditions. So traits, what we observe as individual characteristics, are almost all a combination of genetic and environmental effects. So there's very few traits that are entirely environmentally or entirely environmentally determined or entirely determined by genotype. Knowing how heritable a trait is actually very important because it informs our breeding efforts. So we can't improve 100% environmental traits over time, but we can improve traits which have at least some genetic component. So we can improve phenotype or crop production in general via either optimization also of the environment. This is actually not plant breeding, this is agronomy. And this includes management practices such as pesticide, herbicide, and fertilizer use. Things like sowing time, tilling, intercropping, crop rotations, watering. These management practices in general belong to the optimization of the environment for the crops. And then on the other side, we have the optimization of the genotype, which is producing new cultivars, and this is the plant breeding area. So this includes breeding goals, such as breeding for enhanced pest and disease resistances. We call all traits which result from living organisms, so like pests and diseases, biotic stresses. And then things like drought, waterlogging, heat or cold resistances, and these are abiotic stress resistances, everything which comes from non-living stresses. We also would like to improve things like nutrient use efficiency, quality traits, crop architecture, and this kind of thing. And of course, yield, which is always a combination of all of the above, plus specific yield-related traits such as seed production. Okay, so improving crop plant production actually means that agronomy and plant breeding need to work together. So agronomic practices direct breeding goals. So for example, what are the targets that we need to achieve from plant breeding to produce cultivars which fit the agronomic practices? So do we need earlier flowering cultivars? Do we need to develop, for instance, resistances where there is no chemical control possible? So if it's not possible to develop an agronomic control method, then do we need to go for the genetic improvement? And plant breeding also informs agronomy-based practices. So the crop cultivars which are available actually also direct the agronomic practices which are possible. So for example, like, do we need to spray? Are these plants already resistant? And which varieties should we plant in order to achieve the best yield for the best environmental conditions? And this brings us to another very important topic that I would like to briefly touch on, which is genotype by environment effects. So if everything is a product of genotype and environment, this sounds really relatively straightforward, but we also have the possibility that we have these interaction effects between genotype and environment. And what this means here, I would like to demonstrate with this graph of cultivar productiveness. So we're looking at different environments from very poor environments to very good environments along the x-axis here. And then the y-axis is increasing yield. So we can see here, we have two different concepts I'd like to introduce. One is yield stability, which means that a particular genotype shows similar yields under different environmental conditions. And then we have yield responsiveness, which means if we improve the environment for the cultivar, for the genotype, that it actually also will give us improved yield, so both of these things are highly desirable from a farmer perspective or from a breeding or agronomy perspective. Here, with these cultivars, we have cultivar A, which is stable but has low yield even in good environmental conditions. So this is not really an optimal cultivar to choose. And we also have cultivar B, which is a high stable yield under all conditions. This is what we want, but this is probably a magical unicorn variety. This is not necessarily something we would see. Actually, A and B are relatively unrealistic because most genotypes react differently to different environments. So A and B do not have genotype by environment effects, but we can see for these other varieties here, we have, for instance, variety C, which has low yield under poor conditions, but good response. So if we improve the environment, then we also get improved yield. And this is an example of a genotype by environment interaction. Line D also has a better stability than C, so the yield is relatively similar between the low and high environments, but it doesn't also improve as much as C. So this is actually a worse cultivar under poorer environmental conditions. And we have cultivar E, which is objectively worse than C or D, even though it has a better response than D does. So when we're selecting cultivars, whether we would choose C or D as the best variety actually depends on the particular environment. And this is another example of how the breeding and agronomy practices need to also work together. Okay, so what do we mean by available genotypes? We're usually talking just about crop cultivars. And these are the product of long-term selection processes. So we start off usually with all individuals within a species, and then over time, these are domesticated into our crops, our crop types. And then we have a further selection process. Each arrow is like just representing many generations, by which we get cultivars. So cultivars means sort of genetically similar lines or homogeneous lines, which can then be directly sold to farmers. So cultivars are basically the end product of the breeding process. This takes many generations, so this is not a fast process, but it's been quite successful in producing modern day crop cultivars. The main effect that has allowed us to achieve go from this extremely high genetic variation to start with to these homogeneous higher yielding cultivars is the principle of selection. So selection is simply choosing plants with particular characteristics or traits as the parents of the next generation in order to produce offspring with improved characteristics. So we want always that the next generation is better than the previous generation. As a simple example of this, if we start off with these plants here and we just want to select for taller plants, we can select this individual as a parent for the next generation, and then we can hope that we have an improved height in the progeny. The problem is we always need to consider, because if we have this as our product, then in this generation, no further gains are possible. So if all the plants are the same height, we can no longer make any selective gain. There's no way to select a better plant if they're all the same. So we often have to balance between introducing new genetic diversity and improving our selection efficiency. But so traditional crop breeding and domestication also took us quite a long way. This refers to the larger scale changes we see over thousands of years. For instance, here, this is a very classic example where teosinte is the progenitor species for maize. And we can see very classic domestication phenotypes, which is these much larger fruits or the harvestable part of the plant. So the cob for the maize is much nicer looking than the teosinte. And we can also see this single stem phenotype as well. This is also an example we see in tomato. So we can see the wild tomato on the left, and this has got very small fruits and also this multi-branched structure. And by contrast, we have the domesticated tomato, larger fruits with a single stem and more above ground biomass. A more extreme example then is also when we have the domestication into different crops from wild mustard, for instance, where we have these differentiation into kohlrabi, kale, broccoli, Brussels sprouts, cabbage, and cauliflower. And this is through selection for specific mutations or for specific parts of the plant, basically. So this worked quite well to give us our crop cultivars. But the problem is that this process generally is actually very slow. So crop domestication, here, these are two major domestication traits. So non-shattering means that the seeds stay in pods, so you can harvest them easily. And this is a major domestication trait, because if the seeds drop to the ground before you can grab them, then this means these plants don't tend to be selected for. And also, of course, grain size, which is generally quite important. So you can see here, this is different crops and how they've fixed these traits over time or improved these traits over time. But the important thing here to note is this axis is actually thousands of years. So this is crop domestication, but it's very, very slow. It is selection, but it's not very targeted selection. But yield increases due to modern crop breeding are impressive. So we can see gains here for major crop species, maize, rice, and wheat, going from 1 to 2 tonnes per hectare up to 3 to 5 tonnes per hectare over the course of about 50 years. And this is a result of modern breeding practices. So modern breeding goals are this is not comprehensive, include, as we said before, the biotic stress resistances. We want to introduce insect resistances, resistance to fungal, viral, and bacterial disease, competition with weeds potentially if possible. We also would like our plants to be drought, heat, cold, flooding, wind, heavy metal tolerant. So have basic tolerances to all of these environmental factors. We would potentially also like to select for physiological traits, which we're interested in. So physiological traits are things which are internally, the internal activities of plants. So this is nutrient use efficiency, photosynthetic efficiency, primary and secondary metabolite production, and oil and protein content, this kind of thing. And of course, yield and yield-related traits are always very important. That can include also things like plant architecture optimization, but yield is of course affected by all of the above traits. So it's not the case that even if we have a high-yielding cultivar, but it has no disease resistance, that we expect to have a high yield if the disease is present in the field. So if all of our plants die because they're all susceptible to a disease, we're not actually going to get any yield at the end either. Okay, so also modern crop cultivars are a product of all of these modern breeding practices. And there are many modern breeding practices, so I'm not going to go through too much this in detail, but improved selection efficiency is really key, so based on better understanding and calculation of heritability. Also, like major advances such as the production of pure breeding lines or of hybrid breeding, which takes advantage of this hybrid vigor effect, have made major advances. But what I'd like to focus on for the remainder of the talk is also the development of underlying research fields. So I'd like to just give an overview of how our better understanding of the biology of crop plants, molecular genetics and genomics, quantitative and population genetics, and biotechnology have led to advances in plant breeding. And specifically, I just want to explain what these things are. So I just want to give a very brief overview of these different fields of research and how they potentially impact also on the improvement of crop genetics. So this is really now crop breeding research as an overview. So plant breeding sits kind of in the middle of this research concept mapped here. And we can say that the major contributor is genetics and heredity. This is the most important bubble. But we also have, of course, agronomy, plant sciences, agriculture, horticulture, evolutionary biology, biochemistry and molecular biology, and biotechnology and applied microbiology as fields which potentially impinge or can be related to plant breeding. And I didn't come up with these. These are the ISI Web of Science Journal Citation Reports categories. So this is the number of journals in 2019 which covered each of these fields. So some of these also are not related to plant science. So the journals in biotechnology and applied microbiology, biochemistry and molecular biology, and genetics and heredity also overlap with other fields such as medical or animal sciences. Just to give an overview. Okay, so what is genetics and heredity? The word genetics actually only dates back to 1905. So this is a relatively recent concept, but since then this research field has exploded in different directions. So we have Mendelian genetics, molecular genetics, quantitative genetics, population genetics, a little bit evolutionary biology, and genomics. So I'm going to briefly go through what actually each of these things are. So Mendelian genetics was the start of it all in principle. So modern genetics really started with Gregor Mendel's Versuche über Pflanzenhybriden, which he published in 1865. So this paper described inheritance of traits in pea plants as a combination of discrete factors that resulted in ratios of types of progeny. So Mendel's peas are quite well known. This is where we had these yellow and green seed phenotypes or purple and white flowers. And we have segregation of discrete genetic factors. So this was the antithesis to the blending idea where it was hypothesized that individuals were just like a mix of the traits of the parents. Instead, Mendel worked out there were actually discrete factors which control genetic inheritance. So these Mendelian traits describes these traits which have discrete inheritance. So we see either purple or white flower or something, or not like a mix of colors between purple and white. We now know these are actually traits controlled by only 1 or 2 genes, but this was not known at the time. This is long before the discovery of DNA sequences or of any actual molecular way of conveying genetic information from one generation to the next. But he established that there are these discrete factors which can then be inherited in the progeny in predictable ratios. So we see sometimes these keywords, dominant, recessive, random segregation, independent assortment, or pedigree. These belong to the Mendelian genetics area. It's still important for breeding research today because calculating Mendelian ratios in progeny allows us to determine how many genes might be controlling a trait of interest. So do we have major gene traits like single gene traits, or do we have many genes which contribute to a specific trait that we're seeing in the population? These single gene traits are not the majority, but they're not uncommon either. So the most major resistant genes are basically Mendelian in inheritance. So we still today use these Mendelian ratios in order to work out how many genes might be responsible for a particular resistance phenotype. Linkage mapping, which I won't go into, also relies on Mendelian principles. So this is also very critical for understanding how breeding tools like QTL mapping work. Okay, but Mendelian genetics didn't explain everything or actually very much, so it was a big breakthrough, but there were clearly some issues relating to the inheritance overall of traits from parents to progeny. This led to the rise of 2 new fields, so population genetics and quantitative genetics. And these basically kicked off, so population genetics was first. This is generally considered to have started off with the work of Wright, Fisher and Haldane, working in the sort of 1920s, 1930s. And it integrated natural selection with Mendelian genetics. So Darwin had published these exciting papers about how species evolve over time. So the finch bigs potentially to evolve on different islands to based on the available food supply. If they're eating insects, then the beak shape is different from if they're eating fruit. And also this moth example, we might be familiar with the coloured moth, the selection for particular moth phenotypes based on pollution on trees and predation by birds in England following the Industrial Revolution. So the idea was this underpins sort of the evolutionary biology principles, and the field of population genetics is quite heavy on mathematical modelling. So it uses equations to describe how allele frequencies change in populations. So alleles are just genetic variants. I'll probably be unable to stop myself using this specific terminology, but a gene is just something which results in a trait, and then an allele is just a variant of a gene. So for instance, for eye color, we have different alleles, which would be blue, brown, green. These are alleles of eye color. So keywords which belong to this area of genetics is like selection, mutation, genetic drift, gene flow, Hardy-Weinberg equilibrium, allele frequency, and genomic selection. You might have heard some of these. It's important for breeding research because it's actually the foundation of selection theory. So including the breeder's equation, which gives us a rough estimation of how much we can expect to improve a trait from one generation to the next, depending on how heritable the trait is. So it describes best practices also to get results in breeding using statistical prediction. It's also the basis for genomic selection, which is an increasingly popular tool, which breaks down the information of the genetics over the whole genome into basically models, which are then used. So every marker or every genetic locus or every allele is given a particular value, and these values are all calculated together to see what the effect on, like the predicted effect on yield would be for a particular combination of genetic factors. So the interesting thing about this is you can actually make breeding gains even with no understanding of the underlying biology of traits using genomic selection. So it's really more of a modeling approach. Okay, quantitative genetics is a little similar, but instead it deals with phenotypes that vary continuously, so not discretely as in Mendelian genetics. And this is examples such as height, weight, and yield. So the Mendelian traits or the single gene traits tend to be things which are resistant, not resistant, black, white, yellow, green. So these highly qualitative traits or phenotypes. So we expect category variables. But continuous traits, where we have a distribution in the population, so that can only be described by numerical data, includes most of our important traits, including yield. So this overlaps with population genetics a little bit, but tends to deal more with specific genes. So the attempt here is also to uncover genetic networks and smaller effect genetic loci. So if we have multiple genes which contribute to a trait, we usually are going for quantitative genetics. The keywords which belong to this area, we have polygenic, quantitative, distribution, QTL mapping, additive effects, epistasis, and genotype by environment effects. It's important for breeding research because most traits are quantitative, controlled by many genes, and showing a continuous distribution of values across a population. And it also offers a comprehensive toolbox to allow detection of quantitative trait loci and genetic factors affecting traits. So quantitative trait loci is just when we're talking about lots of genes having an effect on a single trait. We also use this for marker-assisted selection, which is another breeding method, and it can provide a basis for further investigation into the molecular basis of traits. So what genes are actually responsible for a trait in the first place. Molecular genetics is a bit newer, so this is post-Mendelian and also a bit post-population and quantitative genetics. And in this case, this is more about finding out specific genes and their allelic variants and the way these result in specific phenotypes. So this deals with the individual gene level and what we call the central dogma of molecular biology, which is that DNA is the template for phenotypes effectively. So DNA is transcribed into messenger RNA. This is sent into the cell machinery to make proteins. And the proteins then build the organisms that we see for the phenotypes. So the keywords is mutant, allele, tilling, gene expression, reverse genetics, forward genetics approaches. Molecular genetics is actually mostly at the DNA or gene interaction level. And then we also have molecular biology, which is a bit broader. I usually consider this as more looking at protein, gene to protein or protein interaction level. And then this also leads into biochemistry, which is further downstream, where we're mostly looking at the production of biological molecules. So this is kind of the path from the DNA through to the proteins and through to the effects on the traits from molecular genetics through molecular biology to biochemistry. Molecular genetics is important for breeding research because it helps us really validate which specific genes or allelic variants are causative for crop phenotypes. So what is the role of each gene in this trait? We can use it also then to develop functional markers, so markers which allow us to say, okay, that a particular allele is responsible for this particular phenotype. It's also involved in the development of novel phenotypes, so mutation breeding falls into the molecular genetics area, and it underpins a lot of basic biology. So molecular genetics helps us to understand how plants grow and develop and how specific traits are determined at the genetic level. Genomics is relatively new, so genomics attempts to extend molecular genetics principles such as gene expression or allelic variation to the whole genome level. Genomics defines gene networks, so not just how genes interact with each other, but also how genes are expressed. So in response to environment and regulatory interactions which affect phenotypes. So genes, most cells within an organism actually have the same gene content, but some genes at any particular time are switched on or switched off in response to different plant developmental stages or in response to stresses from the environment. And this switching on and off of genes is called gene expression. And this is obviously very much determined by genotype by environment interactions, but also genes affect other genes, and this can be quite complicated quite quickly. So we have, we say we have global gene expression pattern regulation, for instance, or gene networks. And this refers to this organization of which genes are going on and off in response to different developmental or environmental stresses. So genomics also looks at the organization of DNA into chromosomes and chromosomal regions and how this affects gene expression, and also things like 3D structure of DNA. In this area, we have cis-trans regulation, gene expression networks, gene ontology, chromatin conformation, and of course, like sequencing. So next generation sequencing approaches, etc. Bioinformatics is actually the development of tools for genomics analysis. So bioinformatics effectively is the sort of the pure form, and then the application of bioinformatics methods is really genomics. There's a bit of argument over these definitions, so it's not so critical, but this is kind of what we're talking about here, that applied bioinformatics is genomics. Why is it important for breeding research? Genomics is now practically the only method we use to identify genetic variants for genotyping, so for molecular genetics approaches or for genomic selection. And it also offers a suite of methods to expand molecular genetic insights into the actual variants responsible for traits. So it tells us a lot about the genome, it tells us a lot about how genes interact with each other, and then also sometimes about physical factors in terms of the chromosomes, which limit breeding gains. So chromosomal location of genes, mutation rates, fixed haplotypes, this kind of thing. Okay, biotechnology. So biotechnology is a very broad term. So this is just biological technology, and it deals with the exploitation of biological processes for various applications. It's not really clear who decides what is biotechnology and what isn't. This term is very widely used. Conventionally, this was usually meaning tissue culture or genetic transformation of plants and microbes. So the main point is it's a fairly strongly applied or methods focus. And here we're talking also about things like genetic engineering, transformation, tissue culture, cell culture, genome editing, like microorganisms, etc. It's important for breeding research because genetic engineering and genome editing can allow us to directly make genetic changes that produce beneficial traits in the plants, when this is actually allowed by country-specific regulations. Tissue culture also, so that's when we take pieces of leaf or something, for instance, and then actually grow them in a petri dish in the lab. We use this for making hybrids between related species or between distantly related species, and also for making these pure breeding lines very quickly. So not so critical exactly how that method works. Biotechnology also includes a quite interesting area of research, which is turning plants into factories that produce useful compounds like pharmaceuticals or vitamins. But this is usually also classed as GMO technology, and so is not necessarily so common in Germany. Plant sciences is a very broad research topic. There's actually more research funding in medical and related fields than natural sciences. So we have this catch-all plant science kind of group. So this includes many subdisciplines from ecology to physiology and development and cell biology. And it overlaps as well with molecular biology, genetics, and biochemistry. So as well as model organisms, this is increasingly covering a lot of research on crop plants. It's important for breeding because it's understanding basic principles of how the traits work. So how does plant physiology, nutrition, or disease resistance function? This is critical for molecular genetic and genomic approaches to breeding. So we can use this to identify candidate genes responsible for specific traits, to produce markers, to understand the inheritance and interactions of traits, and work out which traits are good candidates for selection. Okay, so agricultural research, this is just from the ISI Web of Science, is agriculture in general is any type of study aiming to improve productivity and quality of cropping systems, including genetic improvement, plant protection, inputs, mechanization, marketing, and research management. So agronomy is specifically is agriculture applied to cropping systems, so excluding animal agriculture. Horticulture is agronomy, but it's actually for fruits, flowers, and vegetables, so anything that isn't a broadacre crop. And I'm afraid forestry research is too small to get its own research bubble in the ISI Web of Sciences. Of course, agriculture is important for breeding research because by many definitions, agriculture includes breeding research. So agriculture is for sure what defines also our most important breeding aims. So increasingly, like classic breeding for applied agricultural improvement is being done in industry rather than by research-intensive organizations like universities. So not to say that the universities are useless, but that it's also, the field is very large for research which is happening in plant breeding. And there's actually also some level of pre-breeding. So this is development of material that may not immediately have applications that's being done by industry, particularly by very large companies like KWS or Bayer, after the recognition of how important this work is in terms of making continuing gains in crop production. Okay, so just in general then, plant breeding research can involve many different fields, so it falls kind of in the middle here. And yeah, generally the main aim or the main tying together for plant breeding is when we're looking at genetic improvement of crop plants. That is fundamentally what we're doing in plant breeding. So you can say you're doing plant breeding if you're effectively trying to genetically improve your crops. Okay, so very briefly, what we're aiming to do with robotic phenotyping data, potentially we would like to have better phenotyping or deep phenotyping. So we want to see if we can measure traits that are difficult or time consuming to measure by hand. So where it's not practical to get the amount of data we need, or we can see how things change over time in much finer detail. Maybe we can use image analysis to identify new traits of interest. It's not the case that we necessarily think to measure everything that might be potentially interesting. High throughput phenotyping is already in progress a little bit as well. So this is with faster, more efficient screening of large numbers of individual genotypes. So this is very important for breeding programs or for in general research that we can get a very quick phenotype analysis. So phenotyping is now the constraint in terms of time for breeding. So it is now very quick to get genotyping data, but phenotyping is now the bottleneck for making progress in progress in crop improvement. And of course, we want to improve selection efficiency to determine which individuals have the best phenotypes. And we also just want to better understand the genetic basis underlying crop traits so we can help this for future research. Okay, so thank you very much. I'm done and I'm happy to have questions.